Good afternoon, everyone. I'm Basil Balsis, and I'm serving as the Interim Dean of the Newark College of Engineering. On behalf of the deans of all colleges, I would like to welcome you to this event. I would like to recognize the presence of our Provost and Senior Vice President, Dr. Dirk, and our Senior Vice President for Research, Dr. Sebastian. Dr. Bloom, our president, regrets the fact that he could not be with us today. He has some other important duties for the university, but nonetheless, he sends his regards, and he is very supportive of this event. As you know, we are in a third year of hiring new faculty, and this is going to be concluding a 3-year plan, according to which the university is investing in new faculty in order to expand the research portfolio and horizons of our university. The themes that have been selected are essentially the interface of engineering and life sciences, the digital transformation that we are undergoing as a civilization, essentially, and sustainable systems being the third one. Through this hiring plan, we have added a good number of faculty, and in this current year, we have added 12 faculty members, 8 of which were hired according to the plan that we are all familiar with, and 4 were hired as replacements of other faculty. All of them extremely talented and accomplished and promising individuals. And this mini symposium is dedicated to them, and it features this new faculty. We are really pleased to have these people with us, these colleagues with us, and we are looking forward to the conclusion of this hiring plan, essentially counting replacements. We are anticipating that a year from now, we are going to be having, over the 3-year period, 50 new faculty, essentially rejuvenation, if you wish, of the faculty at about 1/6 of its capacity, which is really important. So let's start with this mini symposium, which we have chosen the theme of technology and human health. We have 3 oral presentations, and then there are posters outside, and I'm sure you are going to have the opportunity to discuss things with our new colleagues. The first speaker today is Dirk Bucher, who is an associate professor of biological sciences. Dirk came to NJIT from a position of assistant professor at the Whitney Laboratory for Marine Biosciences in the Department of Neuroscience at the University of Florida College of Medicine. His primary research interests are in the area of information transfer in animal motor control systems, with a main focus on understanding how activity and neuromodulators alter, shape, and stabilize neural network function. The talk he's chosen for today's presentation is Neural Circuit Dynamics, Lessons from Lobsters and Crabs. Dirk?
Good afternoon, everyone. Oops. So what I thought I should do is just give you a general overview about why we study, or why we use lobsters and crabs to study neural circuit dynamics without showing you too many details and actual data. Before I start, though I wanted to say, maybe for the benefit of the administration that is present, that I moved here from Florida, and I wanted to come to NJIT so badly that I left this for this. And so there's a reason for this, and this reason is that there's really a very vibrant and large neuroscience community at NJIT and around it, and I have some very like-minded colleagues and actually collaborators in the department, and we all study the brain or nervous system function, and you can do that at different levels, all the way from molecular to behavioral. What we are interested in is the general or very broad question how cellular, neuronal, and synaptic properties give rise to circuit function and dynamics, and how that feeds back to the neuronal properties. Again, there's two challenges here, basically. One is, in most systems, the sheer numerical complexity of circuits, with thousands or even tens of thousands of neurons that are interconnected, and us neurophysiologists can go through them one by one with our little electrode. And the other one is dynamics. And what do I mean by that? Well, let's start with sort of the introductory textbook versions of how neurons and circuits work. So we have a neuron with its dendrites and an axon, and it receives synaptic input. We can stick an electrode into this and measure voltage over time. And what we see are synaptic potentials that get summated till a threshold is reached, and then an action potential is generated that can be propagated and help the neuron talk to other nerve cells. Well, if it was that easy, all we would need to do is figure out what the circuit connectivity between all these different neurons is, and then probably get some pretty good idea what the circuit's going to do. And in fact, a huge effort is undertaken at the moment in the mammalian brain to figure out, or in a better way, how circuits are connected and how different circuits are interconnected. Well, that's not going to give us, I mean, that's an important endeavor, but it's not going to give us the answer how these circuits actually operate. And the reason for this is twofold. One is that there's a huge diversity in both intrinsic and synaptic properties, and there's a lot of dynamics. So synapses, for example, don't usually just summate linearly their inputs, but there is short-term synaptic dynamics. So during repetitive activity, you can get things like facilitation and depression or depression. And the intrinsic excitability of neurons can also be very diverse and very nonlinear, apart from the fact that there's a threshold for firing. Some neurons, for example, are spontaneously active without needing any synaptic input, and they can even produce oscillatory activity. Some may adapt the firing rate to constant input. Some may even fire in rebound from inhibitory input. And there's a huge diversity of these nonlinear properties that are very, very cell type specific. So to make things even more complicated, the ion channels, the ionic mechanisms that underlie these intrinsic and synaptic properties, both the synaptic ion channels and the intrinsic voltage-guided ion channels, are not fixed in their properties. They can actually be changed by neurohormones and neuromodulators that act through receptors and second messengers and change the gating properties of these membrane proteins, and basically can radically alter the excitability and the synaptic interactions between neurons and give rise to very, very different versions of circuit output. And finally, there are long-term regulatory mechanisms that involve things like gene expression and protein expression that can change at a longer time scale and more persistently the intrinsic excitability of neurons and the strength of and dynamics of the synaptic connections between them. So the challenge really is, if we want to understand how neuron and circuit dynamics works, is that we don't just have to know the interconnectivity, but we have to understand the different types of dynamics that can act at very different timescales and that can also really interact with each other. So both for the normal function and the brain, this is important, and for pathological conditions, many of which can be viewed as a failure of having the proper interactions between these kinds of dynamics. Okay, so if you want to do this in the complex mammalian circuits, in the complex circuits in the mammalian brain, I wish you the best of luck. We have chosen a different path, and we are using the stomatogastric nervous system of lobsters and crabs. This piece of the nervous system is sort of an accessory part of the crustacean nervous system that controls rhythmic movements of the stomach. So in a way, maybe the world's most boring behavior, but the preparation just comes with very distinct experimental advantages for the accessibility that we have to the system. So what we usually do is we take out the stomach of the animal. I'm not going to tell you what we do with the rest of the animal. And we can lift this piece of the nervous system off of the stomach and put it in the dish. And it's basically a series of interconnected ganglia, so bags of nerve cells that are connected by nerves. And the central, and we know the details of the anatomy of it, we know which nerve cells project through which nerve and so on and so forth. The central part of the system is the STG, or stomatogastric ganglion, that contains the circuits that produce this rhythmic output that controls the stomach movements. And if we look at anatomical staining for this, on the left side you see background stains that shows all the neurons of the stomatogastric ganglion in green, and then in red the synapses. On the right side you see a single neuron that has been stained with dye through an electrode in green, and in red also the synapses, just to show you the beauty and the complexity of the morphology of these neurons. One thing I want to point out here is it's immediately clear that this STG has a very limited number of neurons. Depending on species, there's just around 30 of them, so not thousands or tens of thousands. And they're very large, so the size of these is about 10 times of that of your typical cortical neuron, which makes experimental accessibility a lot better. And because there are so few of these neurons, people have figured out relatively early that most of them are individually identifiable neurons that we can unambiguously identify across different experiments in every animal, which is a huge advantage because you know that you're not comparing apples and oranges, and it's not just cell types, but it's actually individuals. And because there's so few of them, it's relatively straightforward to figure out the circuit connectivity between them, and we have known this for a long time. So we actually have sort of the engineering circuit diagram for this circuit. Even better, if we take this piece of the nervous system out of the animal and put it in a dish, it spontaneously produces these nice regular rhythmic types of activity without the need for any electrical stimulation or pharmacological activation or anything like that. And it'll do so stably for, if you do it right, up to several weeks actually. And these patterns that are produced here are very, very similar to the ones that we can record from the attacked animal if we do muscle or nerve recordings. So basically what this means is that this piece of the nervous system, even if you take it out and study it in vitro, produces the same kind of output activity as it does in the intact animal. So one of the things that the stomatogastric nervous system is famous for is neuromodulation. And this little circuit is modulated by a large number of different neuromodulatory substances or neurohormones, some of which you have heard of probably, things like dopamine and serotonin, and then a huge number of different neuropeptides. And these neuromodulators are actually essential for the operation of these circuits. So if we do an experiment where we have this isolated piece of the nervous system, you see there these three ganglia on top that contain neuromodulatory neurons that project to the STG in the middle. And if we leave everything intact, we get this nice regular rhythmic pattern. If we block the descending neuromodulatory inputs, you see that most of the time at least all rhythmic activity ceases. So the operation of these circuits to control movements of the stomach is crucially dependent on the presence of neuromodulators. What we can then do is, one by one, bath apply different neuromodulators, and in most cases we see that there's some version of this rhythm elicited by the application of neuromodulators. And there are decades worth of work now and a lot still to be done in trying to understand how different modulators reconfigure these circuits in different ways by acting at different subsets of the neurons in the circuit, and within one neuron at different subsets of cellular targets like ion channels or synaptic proteins and things like that. So obviously one of the perspectives to have on this is that that's not very different than the mammalian or even the human brain operates, and neuromodulators are very, very important in our brain to control functional and healthy output, as you can tell by early stages of Parkinson's, for example, where the neuromodulatory signal of dopamine is compromised. So I'm not going to say anything more about circuit neuromodulation, but there is or at least not directly, there's another very interesting aspect of this, and that is that we can do this experiment in a slightly different way. So here is what I've just shown you. So we have a control rhythm. We can block descending inputs, and the rhythm disappears. And then if we apply modulators, we get some version of this rhythm back. If instead of applying a modulator, we block the descending inputs and we just wait for long enough. Sometimes that takes tens of hours, sometimes just a day or two or something. After the appropriate time, at least most of the time, we get functional recovery. So the circuits are now able to produce rhythmic activity in the absence of neuromodulators that had been crucial before we did this manipulation. So this obviously has obvious implications for things like spinal cord injury, where the question is not just how to do wound healing and get rid of the lesions, but we also have to worry about what happens to the circuits that are underneath a lesion and don't get inputs anymore. And many investigators have looked at this kind of what I would call homeostatic plasticity, including Jorge Gulawash here at NJIT, who has been instrumental in figuring out some of the underlying mechanisms of this type of recovery and what that means for intrinsic and synaptic properties that give rise to that. I just wanted to show you recent research direction that we have taken with this. It turns out that it's not just the ion channels and the synapses that change if you deprive the circuits of neuromodulatory input and activity, but the neuromodulatory mechanisms itself can respond to this kind of manipulation or injury, whatever you prefer to call this. So here is spike frequency as a measure of excitability at different concentrations of a modulator like this. And what you can see is that in black is control. If we deprive the circuit or this particular neuron of modulatory input, the responsiveness to this neuromodulator actually increases. So there's some sort of compensatory action that makes this neuron more susceptible to the substance that you have deprived it of. If you do the opposite experiment, where you block the normal descending inputs but then incubate for the appropriate time in a high concentration of a neuromodulator, the responsiveness to that neuromodulator actually decreases, which you see here in green. These things are accompanied by a change in the expression level at the mRNA level of the gene that codes for the receptor for this particular neuropeptide in this case. So again, this has obvious implications for things like nervous system injury and the basic mechanisms of that, and how functional recovery can be achieved. So I want to switch gear a little bit and as a last piece talk about something else, that is axonal excitability and temporal fidelity. So let's go back, let's step away from these circuits for a little bit and say that all the synaptic interactions have happened and we have a neuron like this that has integrated inputs and now produces some pattern of action potentials. It fires in some specific way, and these bars are just representing the timing of this. So there's a certain rate to this, and there's a certain interval structure that at the single neuron level represents the neural code, right? And we usually assume that that pattern can now be propagated through the axon to distant synaptic targets, and we usually assume, most neuroscientists actually do, that that's a relatively faithful process. So if this pattern gets propagated to the synaptic target, in this case I drew a motor neuron, so the target is a muscle. If nothing interesting happens during the propagation, there will be some delay, but the neural code will remain unchanged and activate these presynaptic sites. Now, if axonal excitability is a little more interesting, and there are nonlinearities in voltage and time dependencies in there, that could mean that a propagating action potential actually changes the excitability, and action potentials that follow can be propagated at a different speed. If that happens, that would mean that the pattern actually changes during propagation. So you end up with a different temporal code at the presynaptic activation site than you started with at the initiation site. And there are a number of extreme cases of this that are associated with peripheral neuropathies. For example, multiple sclerosis or peripheral neuropathies that are associated with diabetes. That can be so extreme that you may lose a couple of these action potentials, so you completely change your pattern. If that's a motor neuron, you can imagine that that may be associated with muscle weakness. If it's a sensory neuron, that could lead to numbness. Or you pick up a couple of other action potentials, so you have hyper-excitability of the axon, and you end up with more action potential that you actually started with. And if it's a muscle, a motor neuron in a muscle, that can lead to cramping, and if it's a sensory neuron, that can lead to painful sensations. So what we have done in STG axons with this for a number of years now is really look at the details of the intrinsic excitability of these axons. We found that there's a relatively complex complement of ionic mechanisms, including voltage-gated ion channels of different flavors and ion pumps. And without showing you actually any real data, it does exactly what I said before. It's not faithfully transmitting the neural code that is generated at the proximal part of the cell, but there are changes in the interval structure. Now, interestingly, and together with Farzan Nadim in the department here, we have an NIH grant to figure out the underlying mechanisms of this and the consequences for coding. But interestingly, we also found that this temporal fidelity is under the control of neuromodulators. So there are dopamine receptors on these axons that change the gating properties of these ion channels. And when that happens, in this case, even at very low concentrations, the change in the neural code is alleviated. So now, all of a sudden, the axon acts very, very faithfully for transmitting this information. And what we're doing at the moment is trying to figure out what the consequences of these changes in patterns are for the postsynaptic response. Okay, so I think these are the examples that I wanted to talk about. Let me summarize and just say that intrinsic and synaptic dynamics, neuromodulation, and long-term regulatory mechanisms function in concert to ensure stable performance of neurons and circuits. And that bridging different timescales of dynamic properties and regulatory mechanisms is essential for understanding neuron and circuit function on the one hand and dysfunction and disease on the other hand. And with that, I just want to acknowledge people in my lab and collaborating lab, the Schultz lab at Missouri University, that does the regulation of activity and neuromodulator dependent regulation of receptors with us, my postdoc lab, Yves Marder, and then the Nadim and Golowash labs here at NJIT. And with that, oh, and I should probably say that that most of this was funded by different NIH grants. And with that, I will close and are ready to take questions, if there are any.
Take one or two questions if there is any at this point.
Actually, I have a question. I'm puzzled by your slide about the recovery. Is it possible that when you separate the source of modulator chemicals, you kind of spill the chemicals out, and then it takes maybe about 10 hours for them to diffuse to reach that space?
So what you can do is actually difill the descending axons and photoablate them, so you make sure that this isn't just due to random release. There's always the caveat, a little bit of that, but it's very clear. You can also separate these neurons and put them into culture. And even the ones that usually are not spontaneously rhythmically active, but in response to synaptic input, in isolation, they become rhythmic after a while. So there's something in these neurons that tells them, you know, you should be a burster and do this wherever you can, even if you have to use different paths and different strategies to do that.
But you don't know yet what's responsible, right?
No.
Oh, thank you.
Okay.
Thank you very much, sir. Fascinating work, and there is no doubt that unless we gain a fundamental understanding of how things work, we cannot eventually make real progress in terms of prevention of diseases and dealing with malfunctions. However, and although we try to understand how things work at the micro level, there is no doubt that we are faced with a number of health-related problems and problems that have to do with the brain, and we need to do something to address them at this point. And this leads us to the second presentation, a presentation which is going to be given to us by Professor Namas Chandra. Dr. Chandra is professor in biomedical engineering, and he came also not only as professor, but also as the founding director of the Center for Injury Biomechanics, Materials, and Medicine. Dr. Chandra's research interests include computational mechanics of materials at various scales, superplasticity interfaces, nanobiomaterials, experimental mechanics, and blast and blunt-induced traumatic brain injury. More specifically, he focuses on understanding trauma, especially brain injury, using engineering principles and biomedical approaches. Before coming to us here at NJIT, he was professor at the University of Nebraska in Lincoln, and prior to that, professor at Florida State University. His presentation today is going to be under the title, Understanding and Treating Traumatic Brain Injury, a Holistic Approach. Dr. Chandra.
Thanks, Basil. I think I'm extremely happy to be here. And also, in terms of Dirk, I'm happy that he introduced the topic of neuronal modulation, how complex it is. And also, I think he said how difficult it was for him to go from Florida to New Jersey. I took the same route, except I'm happier because I came through Nebraska. So I think though it is cold here, it is still not that cold like Nebraska compared to here. So I think there is a progress that way. And today I'm going to talk a little bit about the understanding and treating traumatic brain injury, an holistic approach. The holistic approach comes from the approaches that it really needs both biological approaches, medical approaches, and engineering approaches. So that's why I'm in biomedical engineering department. All right. And I think we have this CIBM cube, which is Center for Injury Biomechanics Research. And some of these facility that I'm going to show you is based on what I had and what we are building here. And I think you can see this. Overall, I think we try to understand shock-induced traumatic brain injury. I'm going to introduce really, I think, the concept of brain injury, why it is critical. And basically, I think we have the ability to generate shock waves that is the result of the shock wave that occurs in the field, very similar to the ideas of IED explosions and the number of returning veterans suffering both acute and chronic damages. And these shock tubes are capable of doing that. And apart from that, I think we also need a very important instrumentations, both in terms of measurement as well as visualizations. The visualization gives us the ability to capture shock waves that occurs in the order of microsecond to millisecond, and hence we need to have a very capable instrumentation. And also we have to have this same approaches can be taken in the ideas of not only blast. I'm going to talk a little bit about this. My whole idea is understanding the blast and the effect of that. And so not just the experiment, I think the modeling approaches has to be integrated. That's where the holistic comes into picture, trying to take from X-ray CTs and build a three-dimensional model to understand the model, how it works. And also, I think in terms of not necessarily just a human model, because we cannot subject humans knowingly, except some grad students, so some of this blast. So I think because of that, we really has to resort to animal models. And overall, I think, including the animal models, essentially we have to actually have both mechanical models that uses the surrogate called the dummy. Also, we use human cadavers. I think I know this. Some of the people, oh, okay, but I think cadavers gives us a geometry that is required, but the shape and size that is required, not the biological issues. We also use animal models and others. So this overall, we are trying to approach this particular problem of traumatic brain injury from a perspective of experiments, modeling, and integration with respect to that. So traumatic brain injury, as many of you are aware, is something like a silent epidemic. Injury is bad. Brain injury is really bad. Brain is what we are, who we are, and what shapes our behavior. And unfortunately, if you have a lung problem, you can go back and replace lung. You have a heart problem, you can go replace heart. If you have problems in your leg, you can replace it. At least today, we don't know how to replace brain. And we are born with the same brain, and once it gets damaged, it irrevocably changes who we are, not affects just us, our family, and the society as a whole. So understanding this and treating this is some of the goals of our center. Now, in the case of injury itself, as you know, for example, there is about 1.7 million people just in this country having TBI. About 4.2 million concussions take place, which we don't know exactly what happens. And in the case of the right side, what you're seeing is basically the mild TBI, where the soldiers are subjected to. And because, of course, the funding mainly came from Department of Defense, we are really looking at the ideas of how the blast causes injury and whether we can do in terms of protect currently and also treat. Those are the things that we are looking at. Now, going back to this, the injury itself can be really divided into three different parts. One is called the ballistics. Ballistics being near Newark, you probably know it's the bullet injuries. Okay? So next is the blunt injury. Blunt injury is when two bodies come to each other, collide, they are the blunt injuries. Blunt injury examples could be in terms of motor vehicle accidents, even old age falling down, or sports injuries. They are all blunt injuries. Blunt injuries two coming together. The last is basically the blast injury. The blast injury is the effect of when the blast takes place, as I said, in terms of IEDs, or if you are in the wrong place at the wrong time when the blast goes off. For example, you had this, the ideas of Boston. When the people around there is actually injuries that takes place, and some of those blast injuries are caused. So you do find, for example, in terms of ballistics, shrapnel is penetrating, and sports concussion is actually a part of the blunt injury. And again, in terms of blunt impact, you also have the motor vehicle accidents or any accident that goes along. And what really happens is in terms of the concussive injury, the injury basically, injury starts as a mechanical insult. In other words, there's a loading that comes along which our bodies are not designed for. We are designed to protect the brain, but not really fall down 15 feet or take a bullet which is coming at 100 miles per hour, or basically, I think, have a blast. So when that happens, basically, we have an injury. When you have injury, we have different types of injuries, subdural hematoma, epidural hematoma, many different types of injury take place. And then at an axonal level, as we talked in the previous talk, the axon by themselves are also damaged. Okay? And as many of you know, if you're just walking by, and that's the type of blast that is encountered every day. And basically, this can happen as from a pipe bomb to a mortar bomb or intentionally otherwise. So basically, I think a bomb, when it takes place, it creates what we call as a shock wave. Shock wave is nothing more than a blast when the surrounding air gets compressed to a velocity that is more than the acoustic velocity, and it presses so hard that it actually passes very fast. That's a shock wave. Shock wave, when you find when you come from outer atmosphere in the rocket re-entry, you find the shock. So those point of shock wave is when you're even far away, even 100 feet, 200 feet, 500 feet away from explosions, you do find that shock, and when the shock goes, and the shock basically travels with the Mach of 2 or 3, which is basically meaning 600, 700 meters per second velocity. And it goes within about a few milliseconds, 7 to 8 milliseconds. But that's enough to damage our brains. And then, of course, if you are inside that, you can also have the shrapnels coming and hitting you, falling down. You do have a combined kind of stuff. So actually, we were, a few years back, involved. Hopefully, the video will play. So this was actually an experiment that I conducted as a part of working with RV. It's actually in a quote undisclosed location. We had about 40 to 45 explosions. What you saw here is basically, I think, you have an explosion, and this is actually our dummy head where we have instrumented, and that particular one is there. And if you look at again, the same way, this is our example. And let's see whether it plays, otherwise, okay. The same explosion now, you can see this is the shock wave that propagated. So that's all really now is using high-speed cameras. What you saw first was a regular camera. This is a high-speed camera. High-speed camera, you can see the explosion taking place, going up and coming down. All this takes place in the millisecond. During the millisecond, you find the shock wave going by. Nothing really happens to you. Okay, I usually talk very loud. But this actually is the type of loading that we encounter. So however, I think we cannot really repeat this for doing the experiments. For example, if you're interested in doing new protection devices, if you're interested in understanding the injuries, it is not easy to do them, and hence we actually develop the ideas of experiments. So I'm just going to show, this is the shock tube, our dummy head here, this is the field. One of the things we are proud of is actually we had many sensors. Those sensors were instrumented, and you can see this actually are the two. This is the laboratory profile. This is our profile that we generated in our lab. Again, it's a different thing. This is a laboratory profile. This is what we can generate in our lab. Again, the same situations. So what does that imply is we have designed a shock tube that can regenerate exactly what happens in the field, which means if I have an animal model, a cadaver, or any other model in our laboratory, we are actually reproducing what happens in the field. Okay? That's a very important conclusion, because then we can do our experiments to regenerate the conditions that are happening in terms of the protections. So coming back to CIBM Cube, from a variety of situations, actually right now, actually we are in GITC building. For some of you who are not there, you'll see us or hear us very soon. And actually in the last couple of days, we had our first blast experiment that was done with very, very small amount of noise and pressure levels. But this actually is one of the shock tubes. This right now in our lab, shock lab, we have it already. And I think this is very precisely the same design that we use in Nebraska. Okay? And whole idea of the center is not only to really use us, but also get involved, as I'll really talk about the Kessler Foundation, UMDNJ, and all other people. Our goal, the motto of the center, is really protect people and save lives. That's our motto. So I'm going to go a little bit of the devices and what the ramification of what we do at the lab and why I think it should be exciting, just not for me, for all our faculty together. One of the things we can do at this particular point is really understand what type of helmets are really useful. For example, when we really started designing helmet for the military, we never really looked at blast as one of the threats. We really looked at the bullets as a threat, falling down as a threat. When the blast was not a threat, then people were worried. So one of the ways to really test whether the blast is a threat is actually really doing this in our blast lab. We really go back and work with the Army, take the latest model, try to put them together, and understand through direct measurements and modeling whether those particular the protection devices as this being done is really valid or not. We have already made some modifications to the latest one. In fact, actually very simple modifications of relocating some of the pads in direct locations, because to redirect them in terms of the injury, in terms of reducing the injuries. And this is a shock tube that was used. And next week, I think this will be delivered, and you can come back and see it in the lab. And the size of it is reasonably large, and I will tell the mechanics why it is, because basically I think in order to produce a pure shock, we have to make sure, I think it is a planar shock that travels, so it has a longer dimensions. It is basically almost a meter by meter diameter, and the square, and then we have a length of approximately 44 feet in length. And again, you can see some of the tests for the helmet. We can do different helmets and actually conduct the test. And again, this can be the only other way to do this is actually go back and do the field test, and which is very, very, very expensive, time-consuming, safety, and other issues are involved. So for example, some of this actual testing that we have done, and hopefully we can see here. So you can see here actually that the whole process took place from 0 to 7 milliseconds. This is the pressure rise that takes place. These are actual measurements. And you can see here the amount that is cost through other experiments. We could see that enough pressure is going inside the brain, actual brain, where there could be damage. We don't know exactly whether it is damage, but we measured the actual stresses inside the brain. So, and the other part of it is also combining the modeling. Taking MRI CTs, direct normal MRI CTs, we can construct, for example, the ideas of model. And not only that, we conduct the same thing with respect to animal models. So when we conduct the test, one of the things we want to always do is conduct the test and validate the test. Validation is very critical because I think experiments are very important, but the validation, for example, we could go back and look at various regions of it and validate it. All right, so in any case, I think basically what we are trying to see here at this point is, if it ever has to play, is that the concept of we can take the MRI CT and then build a 3D model. Essentially what you do is you build one way, build the second way, build the third way. Once you can do it, we can not only decipher our head, but go up to the level of the tissues and sub-tissue levels, and then use it and then compute it and then calculate it and then validate it. Okay, those are the two things we can do. And so the other one, and as I said, the other one we have done is also using cadaver models. Why should we go into cadaver? As I said, basically I think when you do the animal models are good because they are live and they can give us some information, but the geometry is different. The shapes are different. The shock interactions are different. So in this particular cadaver, as you can see, we can go back and measure the actual pressures outside. We put the ballistic gels inside, measure the pressures. In other words, we measure whether the brain really experiences loading or not. So those are the values, the criteria that can then be used to really design things. Is again, the whole thing takes, for example, in 7 milliseconds. So if you just really see it, you don't see anything. But basically, I think in a 7 millisecond, you can see that, and you can see the actual stress measurements inside to see the value of this is high enough to cause injuries, okay? And of course, we have done a lot of work. We actually worked with UPenn, we worked with Harvard, we worked with Duke, and we continue to work with these people to really look at number of these activities. And these are the various types of pressures. These are all a kind of very important parameters. What these parameters tells you is, for example, suppose there is a C4, God forbid, about 25-pound C4, and people are sitting here. We will have a slightly different type of injury from somebody from the back. That's because the idea of shock as a time distance things. The profile will be different. As the profiles are different, the injury will be different. As the injury is different, in order to do the devices, we need to be able to regenerate many of those variables to see when and where this particular injury takes place. So this, for example, the different profile that we can generate in our laboratory is able to generate different conditions in the field, okay? So once you can go back and look at where the survival is, and using the animal models, we can go back and look at some of the things, again, immunohistochemistries and some of this fundamental work, okay? And I'm not going to do into detail. What I think we are pretty successful in publishing some of this work. In the last 2 years, we are able to publish in at least about 13 different journals. Actually, a couple of them after I came here, and these 3 actually after we came here, we were able to really publish them, because I think there is really a novel work, both from the scientific value as well as from technology value, okay? So, and I think I just want to conclude this. This is a group that I had, but we do have a number of groups at this point. We work with different groups here and the centers. And I think one of the message that I want to say is actually the center is not for me. It is for all the faculty. The problem is complicated, as was illustrated by our previous speaker. It is not trivial to understand how the brain works or how the brain damage affects us. It really requires a collective vision and dedication of various peoples. For example, across not just my department or not just my college, across in our, for example, biology, mathematical sciences, computer science, and also I think in terms of other places of medical school. In fact, that is the reason that I'm here. In fact, the ability for us to really solve the problem by collectively trying to go through this particular work. And I really invite the people who are here or otherwise, just feel free to come to GITC, just to make sure that there is not too much explosion that is going on. But at this particular point, you'll be able to see, and there is any way that we can interact, we'll be more than happy to do it. At this point, I also want to thank 2 people who have been very instrumental. Basically, Don and Fadi has been very instrumental in supporting it from a very short time. We were able to rebuild this. And one other person I really want to thank is Joe Tartaglia. He's not here, but I can tell you one thing. He has been an excellent cooperative person who helped us really establish this lab. Truly, I think in a very short time, and in a few weeks' time, we'll be able to really reestablish where we are and go beyond where we are at NJIT. I believe with this, NJIT can really become the focal point of traumatic brain injury in the tri-state area. That's what I'm looking for. Okay.
Question for Dr. Chandra? Understanding things at a fundamental level, trying to protect ourselves and coming up with proper gear when accidents or other events occur. But nonetheless, even if we are successful in all these things, there are always going to be situations where we would have to go to a medical facility to get treatment. And this leads us to the third and final oral presentation for this afternoon, which is going to be presented by Professor Maria Hurtado de Mendoza, who is an associate professor of architecture. She came to us from the Polytechnic University in Madrid, Spain. She has been twice visiting professor at the School of Architecture at Cornell University. Professor Hurtado is an international award-winning architect and she's also the co-founder of Studio Intrinsic. Her teaching and research examines mathematics and idealized systems of order in architecture. She has been published in major professional and architectural journals, as well as she has 2 books. And she's going to talk to us today about 3x1, variations with repetition, small-scale healthcare architecture. Professor Hurtado.
Hi, hello everybody. Well, first of all, thank you very much for inviting me to be part of this panel. It is my pleasure and my privilege, of course. My name, and now in Spanish, is María Hurtado de Mendoza. Sorry, I know it's difficult. So I come from Madrid, Spain, and I'm an architect and an educator. I'm principal at Studio Intrinsic, which is a research practice, and prior to NJIT, I combined, as is being said, my work as practitioner with teaching at Madrid School of Architecture for 15 years. My field of expertise is architectural design. And this is not only, and yet, a patriotic moment. This is the traditional flag party at the happy ending of a structure without accidents. And I'm showing it because I am a builder. I've been trained to negotiate with the real, always seeking for better translation of abstract conceptual ideas into a physics space. And that is the base of my architectural research. Aristotelian thinking tells us why science, by definition, is abstract, because it aspires to the generality of a law, a relationship, or a concept, and not the uniqueness of an existence. Architecture, in its particular way of thinking, is moving constantly between general and singular. And as you would understand very soon, it may be a non-linear logic. Today I will share with you this back and forth trip in the format of a trilogy of healthcare centers that I have built in Madrid with my practice. And 3x1 is the story of why is that we have built 3 times the same building. It all started with an unusual competition for doing 2 centers for the same client, which was Madrid City Council, with the same budget and the same functional program, but only 2 different sites that were indeed quite irrelevant environments. So our answer to those initial conditions was to work with the idea of a placeless building. A great sense of autonomy was required, either formal and functional and even conceptual, to allow the building develop no matter where. Let's start with one idea set out by Le Corbusier as a reconciliation of opposites, represented by this drawing, a hybrid between sun and Mendoza, meaning that opposites can reach a certain point where they are no longer one thing or another, but both things at the same time. We are interested in the kind of estrangements that comes along with these ambiguous pairings. The so-called placeless building deals with the opposition hermetic open. This is an early model of the roof seen from below, showing the project expression in terms of form and how inside there are different heights and halos. But let's leave it there for a minute and start the same conversation from a different angle. This is a sample of healthcare building plans we have worked on, all in the same scale. When dealing with demanding programs as healthcare, we introduce the concept of programmatic bars, which are stripes of program, chain line configurations of different but similar elements that could be exam rooms or offices or waiting rooms and so on. Looking closely at those bars and having in mind the need of natural light for a good healthcare environment, we can see the evolution of our approach from left to right. First, one base structure where the waiting area is simply the enlargement of circulation and rooms sit by each other to the opposite facade. In the middle, a moment where waiting areas is a specialized part of circulation due to rooms configuration, and also one base structure with a need of 2 parallel facades. Third on the right, courtyards as rooms in a pattern which bring light into each other of the adjacent spaces, regardless function, with no required facade condition. The 3 healthcare centers have the exact same floor plan. The program is disposed extensively on a single ground floor. Defined as a field condition, this is an isotropic disposition that takes the light only from above. We can talk about programmatic atomization rather than sectorization. 3 main conditions arise from that disposition: the public space in orange, the perimeter of the outer shell, the green one, voids and volumes in the shell, the one in the right. This shell is a hermetic and heavy image to the exterior that encloses the open and light space of the interior to emphasize its value. There is a non-stop visual continuity across interiors and courtyards that provides space enlargement. So what you see inside is actually larger than what you can walk through. And so public space is extensive and non-hierarchical, and the corridor vanishes in a certain way. Courtyards bring the light inside the building and help to create a spacious and luminous interior. The transparency and mirroring qualities of the glass also creates multiple visions by reflected symmetry. But I told you this was a trilogy, and in this case, we think that it is possible to read the interior space as a more horizontal and abstract one in comparison to the previous. Replacing the opacity with translucent materials, the result is even more atmospheric. This image also represents for us the creative understanding of evidence-based design concepts, such as the desire for natural light and privacy. We advocate for synthetic thinking as the ability of architecture to solve at once as many problems as possible, including the aesthetic ones. A great deal can be said about what varies and what stays the same in a conceptual way. It is the case of philosopher as Deleuze in difference and repetition, but also topic for linguistics and mathematics. Let me show again what happens in space. Dark blue in the pathios create a vertical relationship and fixes their placement in space as identifiable entities within a larger scenario. Here, the only difference indeed from the previous is the white ceramic tiles instead of the dark blue. But see how the difference between in and out is not that clear anymore. So it's like somehow blurring the boundaries, and the vertical condition is less evident or strong. Third derivative of the same disposition, also very different space condition. And maybe it's just me, but I see like a blast here. Actually, it's X, Y, and Z, or C, as you may say, are more balanced in this image. In this compact geometry, as in many multicellular clusters, perimeter shape is not really what matters. Different perimeter would have not made a difference. It is the connections between pieces what does matter. Very much a bottom-up configuration. And apart from being described as a field condition, the building can be also described as a box, but quite a specific one. The roof plane behaves as a sensitive membrane and becomes really the field facade. It's how building breathes. The absence of hollows in the vertical walls of the exterior enclosure causes the relationship between the interior and exterior of the building to occur vertically, almost with the sky above. Volumes containing double heights and service rooms emerge of the roof plane, reflecting light to the interior. And I would also like to introduce the idea of non-standard. Non-standard appears when varying from the usual. This solid and heavy facade is conceived as a continuous windowless mass. The idea of heaviness is reinforced in the construction by the rough texture formed by the horizontal wood plank of the concrete formwork. As simple as they look, these are the elevation drawings of the concrete walls to be built. The construction system was based on the use of standard forms, so we decided to use only one panel size and to module the whole building. And of course, there were places where the module did not match, but it was important for us to align panels joints with the elevation cutouts, so we wouldn't have uncompleted pieces or pieces with a bite. We added wood texture to the wood planks, and for the no-matching parts, we decided just some adjustments, trying to hide difference, but not placing them in the cutout points. Altogether, we have a non-standard rough skin made out of standard components. And this is a very close look of its materiality. As a result of the whole skin process, we like to talk about identity. And whatever number of different components or parts a project might have, we tend to erase their independency in favor of the wholeness. Like the hand-to-arm continuity, there is no clear division, like here, you know, this is hand and this is arm, and there's no clear line like when one starts. That is probably why we work a lot with the fold-out drawings of the outer shell, like this one, that brings the idea of continuous wrapping or skin. And skin can be enclosure and structure at the same time, or just a thin and golden layer to hide what could not be shown. Maybe why not with a little extravagance here? Or glazing, so the original hermetic image is not a heavy presence anymore, but a disappearing lightness by reflection. We investigate the way to cut in pieces in a discrete manner for things to look complete when joining the pieces back in the construction. And back to Le Corbusier and the system of proportions modular that he defined, by using Fibonacci sequence, modular is the result of merging two views of the same reality, which is man, from geometry and from algebra. Like the two sides of the same coin. From one side, geometry is related to plastic intuition and to aesthetics. From the other side, the numbers game, the rigor. Gregory Bateson, in his book Mind and Nature, also gives a good example for understanding the role of mathematics in the projective logic and its translation into architecture space. He says that every schoolboy knows this equation, but many schoolboys do not know the geometry behind. That is, a collection of four independent but somehow related parts that are contained in a greater whole, and also the idea of length and width that implies. The blue tiles here are arranged in lines with a third of a piece displacement from the one below, so we don't have so much of the static sense of a grid, but a texture surface that expands. To solve the cutting joining issue of the golden skin, we worked initially with a spiral net formed by proportional rectangles. By the way, if anyone in the room knows the name of this spiral, I would love to know. I've been looking for it everywhere. And we thought of cutting out figures from the pattern like this, as if we were using stencils. And again, this idea of virtual expansion and not framing. We were quite excited, but even though the pieces were all rectangular, at the physical execution and placement seemed too complex, so we couldn't afford delays at this moment, and we saved the pattern for the future. Instead, we used the mathematical rule of greatest common divisor. There is only 3 groups of related dimensions, so we could have a free sequence of panels that align vertically, and only at certain moments, whenever they sum the same, they align. And so B equals to A, and C equals 3A, and then C equals A plus B, or 2B equals A plus C. We wanted the mesh texture to go over the paneling without interruptions in pattern. And therefore, panel dimensions is always a multiple of the pattern module, which is 11 by 5.2 centimeters. And once built, maybe because of mathematics or maybe not, Sang gave us this present in the form of spiral. Villaverde's glazing works with the same rules in different proportions. 3 different widths of panels with a greater common divisor, and the alignment every time they sum the same. And the building, somewhere between use and sensation and geometry. In our work, we try to enable the coexistence of day-to-day situations, such as healthcare program, with the extraordinary of its disposition. Creativity as in questioning the given status quo, and from there, the role of the creative in the architectural research. And let me finish by saying that I believe and therefore hope that what I think and what I say as do as an architect and what I teach to my students is the same thing. Thank you.
this concludes the first part of today's mini symposium. Let's thank the three speakers once again for very interesting, stimulating presentations. I would like to invite you to go outside and look at the posters that the other faculty members have put together. I would like to once again welcome all new faculty members and wish them the very best for their career here at NJIT. I'm sure that their presence is going to enhance the work and the output, the productivity of our place. Thank you very much.